This is a screen capture video for Unit 10.3, and on this portion I want to talk a little bit about crossing over. What sometimes happens when homologous chromosomes are close to each other in meiosis 1, in the prophase and the metaphase, there will be a crossover event. I said prophase 1 and metaphase. It's actually prophase 1 of meiosis 1 is where the crossover event is thought to occur. It's when the homologous chromosomes come close together. The chromatids, one on each of the separate homologous chromosomes, can overlap and detach and reattach. It looks something like this. It's easier to show the illustration. You see the two homologous chromosomes. One came from dad, one came from mom. What can happen in prophase 1 when they're close together? The ends of the chromosomes can lap over each other. And this can happen anywhere between the end of the chromosome and the centromere. It's more likely that it'll occur out towards the end, and the end material will be exchanged more so than closer to the centromere. When that happens, the ends detach and reattach, but when they reattach, the end of one chromosome attaches to the other chromosome, and the other end attaches to the other. Here I'll use the colors. You've got the red end that attaches to the blue chromosome, the blue end that attaches to the red chromosome. So eventually, after meiosis is over, these chromosomes have split apart, they've moved away from each other, and then eventually the sister chromatids split apart, and you have 4 different chromosomes. Whereas before, you would have had 2 different types of chromosomes. 2 would have been blue, 2 would have been red, and the 2 red would have been identical, the 2 blue would have been identical to each other. But here you have 4 different chromosomes, and because the genetic material has recombined and formed a different type of chromosome, different information on it than the other chromosomes, these are called recombinants. Now the others are still the same, they haven't changed, but the ones that have exchanged genetic information, those are called recombinants. And this is crossing over. You can understand why it's called crossing over. And where they cross over is actually called a chiasma, this crossover location. And that increases genetic diversity. Something else that should be noted, it's an error in meiosis. It can occur in meiosis 1 or meiosis 2, and that is a process called non-disjunction. And that's when the chromosomes line up along the equator, and one chromosome is supposed to go to one end, the other chromosome is supposed to go to the other end. For some reason, that does not happen. And in this illustration that's showing in the screen capture, you can see 2 different chromosomes that have split apart. They're going to opposite ends like they should. That's proper disjunction. The red colored chromosomes are not moving apart. One should go to one end, one should go to the other. What happens, the 2 chromosomes go to one end, and no chromosomes of that type of chromosome go to the other end. So the cell goes ahead and divides, and what happens, one cell is deficient of one chromosome, and the other cell has an extra chromosome. And this is called non-disjunction. Most non-disjunctions would end in death for the individual. There are some where the person can survive. I'm going to give you some of those exceptions. With autosomes, that's the non-sex chromosomes, the most popular non-disjunction that we can think about would be where there's a trisomy 21. And you can see it right here. There are 3 21st chromosomes when there should only be 2. This is one of the few non-disjunctions of an extra chromosome where a person can survive. But with trisomy 21, the person will generally be mentally retarded, and they'll have some physical abnormalities and usually a reduced lifespan also. We understand from statistics that after the age of 30, the odds of a mother giving birth to a Down syndrome child greatly increase. And at about 40 years of age, it's 1%. And if you look at the graph up here, by the time that a woman is 50, it's about 8% chance that she'll give birth to a Down syndrome child. Here is another illustration of non-disjunction, and this is showing in meiosis 1 where it's occurring. So those are non-disjunctions with autosome, the 21st chromosome. There are some non-disjunction events with sex chromosomes, the X's and the Y's. And there's a chart from the text that shows, it's a little bit hard to see perhaps here, but there's what's called Klinefelter syndrome. That's where you have a male that has an extra X chromosome. That's where there's a non-disjunction. The X chromosome gets pulled over, and there are 2 X chromosomes and a Y. The presence of the Y chromosome determines that the person is a male, but having 2 X's causes this person to have some female-type characteristics. One is usually enlarged breasts. Body shape is a little bit more female than male, and the gonads may not be properly developed, and there may be some mental delays with the Klinefelter syndrome. But you can tell from the illustration here, 2 X's and a Y, and that's non-disjunction of the X chromosome. Another type is what's called Turner syndrome, and with Turner syndrome, the person would be a female because there's no Y chromosome, but instead of having 2 X's, there's just 1 X. There's a non-disjunction where the sex cell did not get an X chromosome, and with just the X from the other parent, the person will have webbing on the neck, and that's one of the key characteristics, and the body shape is a little bit different than normal. Those are 2 of the more common sex chromosome non-disjunction events. There are some organisms that are polyploidy, and they do very well. In the simpler organisms, plants being one, the extra chromosomes don't seem to cause as many problems as what they would in higher forms of life, especially humans. A great example is wheat. Wheat is an example of a polyploidy plant, and that means multiple chromosomes, multiple sets of chromosomes, and that allows for greater genetic diversity and to move in helpful genes that will help the wheat plant to be more successful. That pretty much brings an end to the screen capture, the videos. I hope that you do well on the test, and that's it for Unit 10.